So, welcome to the lecture which I call as interactive session number 10 of environmental geotechnics. And since last few interactive sessions, you must have realized that the central focus is on waste utilization, management and valorization. We will continue with this discussion in today's discussion also, and you must be realizing a lot of possibilities have been created in the process of interacting with all of you here and those who are offline, and these possibilities have to be converted into realities. And the part which is missing and which I have been overemphasizing is that we have not done much in terms of policy creation related to the management, handling and utilization or valorization of the waste. Just as a recap, we have been talking about municipal solid waste and the industrial byproducts, and I also added a tag that in today's world there is nothing known as waste, this is man-made resource. So, this has been the central theme, how to utilize any material which is lying unattended to create value out of it, so that we can go ahead with the sustainability goals, SDGs which we call. So, in this context, I would like to invite Sakshi, who is a second year BTech student and would like to share some thoughts related to what she would like to say regarding the soils.
Thank you, sir. Hi, everybody. I'm Sakshi. I'm a second year student from Civil Engineering Department, and currently I'm a part of a tech team on campus. As a part of the tech team and the competition, I recently went for the International Rover Challenge. My tech team is Mars Rover team, where we build rovers for extraterrestrial travel, and also for one of the main missions in our tasks is life detection. We all know life detection, it's one of the main tasks an astrobio mission can have, since it allows us to know not only how life evolves and arises, but also the possibility of life on other planets. It is the core of space travel. So we have designed a rover to be able to conduct those tasks with ease.
Want to discuss?
Yes.
Go ahead, please.
So on a rover, we start off by identifying the sites where we would like to test soil. We do that with the help of stratigraphic profiling. Now we on the rover, we've just started using this method this year. It's a very amateur level stratigraphic profiling, where we take high resolution images of the soil and the surroundings with our onboard cameras, FPV cameras, and we analyze them using an in-house made machine learning algorithm, which we continuously feed information to when we are not on competition. We have information about the types of soil that we can find, about the information gleaned from different strata of soil that we can picture. We also can capture panoramic images on the rover. So one of the main things that we can get is color variations, grain size and sorting, since that can tell you about the presence or absence of running water, and also the layer thickness. This is the module we use on the rover to conduct chemical tests on the soil collected from the sites that we find favorable from initial stratigraphic profiling. So we have a box with 4 test tubes inside. One of the test tubes also requires a heating component, so we have made provisions for that inside the box. And the main 4 tests that we do, we start off with the pH test. This tells us a lot about the soil. Extremely acidic or extremely basic soils cannot harbor life as we know it, and this tells us also about the composition of soil itself, so we know what other markers to look for. Then we go on with the Bradford's test. Proteins are very essential for life. They're complex macromolecules. They are necessary for life growth and sustenance, so this is why we chose to go with this test as the next marker. We use Kumasi brilliant blue dye, which changes color from brown to blue in the presence of proteins. The intensity of the blue color corresponds to the amount of proteins in the sample of soil that we have. We are planning to include a spectrometer on board to correctly analyze the amount of protein and the amount of
This is a
DNA that we
classical
get.
test which is normally done to find out surface area of soils,
Yeah.
which is known as MB test, methylene blue dye test.
Yeah.
So you take a soil sample and then soak it in MB dye, which is a violet color dye, and then you find out by using a photo fluorescence meter or something what is the intensity of the blue color which has got absorbed on the surface. Clear?
Yeah.
So this will also give you the same n lambda value. And then from there you can find out what is the absorbance of a dye on the soil particle, which has something to do with the surface of the grains.
Okay.
So in soil characterization, the first thing is, you are talking about the protein is very good, microbes and all. Surface area, because surface area makes soils very, very reactive. Correct? So for that matter, if you have clays and if you have sands, which one is more reactive? Clays, by virtue of their very fine particle. So surface area is very high. There are several methods of finding out surface area of the clays or the soil and minerals. And this is where a lot of people are working in discovering new minerals. Fine.
Yes.
And then we have different type of, dyes also, different types of techniques which we will discuss maybe after your mid-semester exams. One thing which strikes to my mind is that when you are taking photographs, what is the resolution of these photographs? Because see, only color change is not because of the moisture. Got it? So there is something known as the water beneath the soil mass, and there is something known as the humidity or the vapors of water above the soil mass.
Yeah.
So if the soil particle is very reactive, what it will do? It will absorb a lot of vapors. Got it? So the moisture presence which you are seeing and trying to differentiate due to the presence, due to the color change, might be misleading. So you will study in geotechnical engineering the role of capillarity. So because of very fine particles compacted, water table rises and soaks the entire soil. That also might result into color change. So it's a very interesting idea. Like I have created more debate out of it for you to realize what you are doing.
Yeah.
Okay, go ahead.
Once we have the positive reaction for the Bradford's test, we go ahead with the DNA test with diphenylamine. So diphenylamine reacts with the deoxyribose in DNA to form a brilliant blue color solution again. This does not react with RNA, which is a failing on our part. We do not have a better test to test for DNA. But since we look at DNA, it is essential to test for life. As we know it, it contains genetic information for everything, which is why we chose to get this test.
Very good. So unknowingly, you have prompted Mrinal. You have some comments on what she is doing, in what way it is connected with your work?
Yes, so this is an interesting thing what you are trying to see. Like we are also trying to understand what are the chances of growth of microbes or presence of growth of microbes in landfill mine soil fraction, and with exposed to different temperatures, how these presence of microbes or will be worthiness of the soil for growth of microbes will be varying or changing. But one question is like, what is the ambient temperature of Mars, means at the surface of the Mars?
Yeah.
Yes, means
You have to give her idea so that she thinks more and more.
Yes.
Yeah. So it's okay, fine. Like the methodology is same. So what we are trying to do is, we are trying to, it's okay. You remember that once you try to remove the landfills, you are doing biomining.
Yeah.
Now the question is whether this soil can be utilized for agriculture purpose or not. So I have to conduct all these tests there. Why? Life. You are talking about soil life. I am connecting it with this material being utilized as a material for as a manure or as a soil rejuvenator or as a soil amending agent. You know, you add some urea or manure to rejuvenate the soil. All right. So the concept is same. So I think you should talk to him and do some tests. Another thing is diphenylamine, which you are talking about, is another test which is known as EGME test. So it's a very standard protocol for testing ethylene glycol monoethyl ether. Correct? EGME. So this gives you what is the surface area of the particles of the soils total, external, and what is exposed from inside also. Like if you take a football and cut it. Got it? So now you have created two surfaces, outer and the inner one, with some thickness of the matter. Correct? So EGME gives you, because it has a lot of penetration inside the grain, which is known as chemisorption. So your language you can follow easily. Yeah, so what comes to your mind now?
Yes.
Can you give her some work?
Yes. If she is interested, then means we are trying to see what is how microbes can harp on the certain materials.
We are now procuring all these bio testing kits for the soils, and our idea is to create a catalog of, what type of microbial species are present in the material which is otherwise lying unutilized and which is getting decayed over a period of time. I hope you will agree with this. Yes, please continue. That's what I thought that she should be presenting in the class.
So the last test that we look for is the XGT test, which tests for cell viability. In the presence of active enzymes in cells, the XGT, the trisodium salt, converts to a highly colored orange formation dye. Again, the intensity of the orange color is directly proportional to the number of viable cells in the sample, and if this test gives us a negative reaction, then we characterize the salt sample as having extant life, and if we do get an orange color, then it is extant life.
So can you perform these tests on the material which we will give you?
Yes, sir.
And then take standard soil from our nursery, from our garden, anywhere.
That is our
And then
testing
compare.
protocol.
Yeah, that's right. So you need not go to Mars. You can create a Martian soil here itself, correct?
Yes, sir.
Any soil which converted to agricultural amendment will serve this purpose. Is this okay?
Yes, sir.
Okay, go ahead.
Again, stratigraphic profiling, that is what we do before, and chemical testing, which is what we do after. We also have a drill.
Just 1 minute. So XTT test, there are some protocols? Have you come across this XTT? Basically, they were known as bioassay tests.
Yeah, XGT is...
Bioassay.
Yeah. It is an assay, and it is highly sensitive, so we don't always get a reaction on this. Because we do need a cell activator and electron.
But yeah, you can find out the presence of the cells in the soils. Very good. So dredge sediments which are of marine origin, from lakes, from water bodies, reservoirs.
Yes.
It's a big challenge, to clean up all this system. Otherwise, you can't store water. Imagine your Powai lake gets silted. So what is the storage capacity? Nothing. So what you have to do, you have to desilt it. Bring this desilted material on shore. What you are going to do with it?
Aggregation.
This is civil engineering aspect. Fine? Okay.
We also have a drill that drills up to 10 centimeter below the soil. We brought it there for the competition to show them. I don't have it right now on me. So the drill collects soil from the samples, and on the site which we feel has the best potential, as per our chemical test, we store soil from there in a cache box aboard the rover, and we bring it back to the site for further testing.
So these rovers are testing using X-ray or some other types of waves?
So
are they doing X-rays, rovers?
we cannot, X-rays are
Or
expensive and out of our scope
or
right now.
ultraviolet or IR, or what is that?
We do have UV
These are the three
sensors.
techniques.
Yeah.
So sensors will either emit X-rays, which is difficult. IR is very easy,
Yeah.
infrared,
We have IR sensors on the rover.
IR sensors. So we now have in our laboratory IR, this thing weighing balances, wherein we can use IR technique to find out the moisture content of the soil. Okay.
So this presentation is more geared towards the technical aspects of our rover. And finally, we do have further improvements that we would like to make, including further testing so that we can have a proper visual of the amount of absorbance of dyes, so that we can have something to compare our results on site with, and optimizing drill designs to have a scooping option in them
So
too.
what is the mineralogy of Martian soils? Is this similar to our
Iron.
soil or they are
Iron
different?
rich.
Iron rich.
Yes.
So my industrial wastes are iron rich. All mine tailings,
Yeah.
is it not? They are iron rich. So truly speaking, there is no difference,
Yeah.
because the tailings which you are using from the mining operations are also full of iron.
Yes.
Fine. Yeah. Okay.
Thank you, sir.
That's it.
Yes.
Thank you. Good job. Very good. Keep it up. But then convert it into a reality. Yes. Yeah. So another interesting presentation which he is going to make, Mr. Yash, on the alkali treated fly ash. So fly ash is also a sort of a waste product which is coming out of the thermal power plant.
Hello.
And there is a major issue that what to do with the ash. But this used to be a very big problem few years back. But now there are rules, norms, and it has been accepted as a construction material. All right, this is already in practice. Incidentally, Hiranandani, most of the buildings were done about 25 years back by replacing up to 32% fly ash with cement. This was a classic example. Fly ash was used as a pozzolanic material. Pozzolana, I think you understand. So pozzolanic material is the one which when interacts with the humidity and water solidifies because of the presence of excessive calcium oxide in the free form. Fine? It acts as a, yes, binding agent. And for finding out whether a material is pozzolanic or not, what you have to do is you have to do XRD, X-ray diffractogram, and XRF, X-ray fluorescence studies, and then you can find out what type of minerals are present and what is that they do. Yes, please go ahead.
Hello, good evening everyone. I am Yash Tarane, first year PhD scholar under the guidance of sir. So this is my MTech project which I have done in NIT Rourkela. So currently I am in first year, so I am going through the literatures of my new project that is related to the carbon capture, utilization and storage part. So basically I have briefly introduced about my MTech topic to you. It is the assessment of alkali treated reinforced fly ash as a subbase and base material for pavement design. What I am trying to do is the full replacement of the natural aggregate. The whole layer is made up of only fly ash and plastic fibers. So I am trying that. So basically there are what I am trying to do at that time is that firstly I have to do some experimental test, and by that result I am trying to make model of the roads pavement by IIT Pave software. Basically divided into two part, if the volume is greater than 2 MSA or it is less than 2 MSA. So there are two different approaches to deal with it. So firstly come to the experimental part. The most basic thing is the geotechnical properties of the fly ash that I use. So what is fly ash? It is a coal based IBP, and it have two types. One is class C fly ash and class F fly ash. Class F fly ash having a low content of calcium oxide, which is, that's why we need some extra agent to use it as a pozzolanic material binder. So for that I am using a NOH, and the main mechanism which going on this process is the geopolymerization.
So what hydroxides do? Hydroxides basically etching glass. All right. You do etching on the glass, is a painting. On a glass sheet, you use hydroxide and dissolve glass and then make a painting, correct? Laser cut also does the same thing. That's laser cutting. Now you can use hydroxides to dissolve the glass and create a painting. It is etching process. So by virtue of their fundamental characteristics, most of these grains of fly ash are silica, quartz. All right. And quartz, when reacts with hydroxides, it will etch and release silica, and silica is nothing but the silica gel, and silica gel is nothing but the gel which you create when you create concrete. So the more and more gelling process occurs, there is a better bond formation. This is the whole logic.
With,
Okay.
with time the gel get solidified and crystallized in form. So these are the basic the UCS, STS, CBR, durability test, permeability and sieve analysis what I have done. It based on the various types of codes IRC that had given to us. Based on that,
Just a minute. SEM is the scanning electron microscopy.
Microscopy.
If you really want to see how the particles look like, and now there are latest versions of SEM also which are available in our institute. We call them as FEC sediment, FEG, FEC, field emission, SEMs, where you can find out even the composition, mineralogical composition of the material by doing SEM analysis, and you can see the texture, grains, how do they look like, whether they are opaque, transparent and so on, fine. And what are the different types of, I will be discussing this later, what are different types of particles which are present in the system and what is their matrix. Yes.
so these are the different guidelines that IRC, that is Indian Road Congress, have published. So basically it's based on the strength parameter and the durability parameter. For any material to be used is as a sub-base material, we have minimum strength of 1.5 MPa, and for base material it should be 4.5 MPa, and the durability of the weight loss by freeze and thaw and wetting dry cycle should be less than 14% of the total mass.
Okay. Yeah.
Yeah. So basically this is the main chart flow diagram or pictures that I used in the IIT PAVE software. So most of the thing is related to this part where the tensile strain and the subgrade is vertical strain. These are the two important finds. These are based on the rutting criteria and fatigue criteria. Based on that we have find the strain, tensile strain and compressive strain by some empirical formula given by IRC, and by that IIT PAVE model I get the values, same values, and I have to compare it. If the value is less than the empirical formula, then it is safe, and that is not, then we have to increase the thickness of the pavement. So what I have done in this, so this is the result. In this basically I have fixed the subgrade of the soil, that is 7% CBR, and fix the traffic volume also, that is 50 MSA. After that I have compared with the different types of pavement. One is granular base pavement, cement treated pavement and alkali treated pavement. So for the granular base pavement the thickness is around 700, and for the cement treated pavement the thickness of subgrade subbase is 150, 100 and 100. For material that I have made, it's about the thickness of subbase is 145 and sub base is 140, and there is one crack relief layer also to reduce the direct impact of the load of the vehicles. So these are the result. Yes.
Okay.
Thank
So
you.
incidentally, what he has done is known as geopolymerization. So in case you get a chance to go to Rome, and most of the buildings in the historic buildings in Italy, they are all geopolymerized. No cement was used. What is geopolymerization? You take an activator, sodium silicate, and then mix it with some siliceous material where silica is present too much. All right, and then leave this for some time. What is going to happen? Geopolymer will get formed. I think who has worked on geopolymer? Yes, Dr. Surendra was talking about geopolymer. So these are known as green concrete. And look at the durability. All those structures which were done so many years back are still standing there perfectly all right. Colosseum is a big example of, geopolymeric structure in Rome. So please, whenever you are there, don't miss it out. You should go and see how this whole geopolymerization was done. In the present day context, people are trying to utilize red mud and sodium silicates and some fly ash or GGBFS. GGBFS is the blast furnace slag. So this is another recipe which is being used for creating paved surfaces, concrete and so on. A lot of applications are there. And it is supposed that geopolymers which are being created are resistive to the extreme climatic conditions where you have a lot of chloride and sulfate ingress. So for offshore structures particularly, they are supposed to be a boon. Any questions? Is this okay?